There's this huge group of bacteria living all around us. I'm talking in the soil under our feet, in our tap water, and they're often completely overshadowed by their one really infamous relative. Today, we're going to pull back the curtain on the other mycobacteria, the non-tuberculous mycobacteria, or NTM for short.
Right? Everyone knows about tuberculosis. It's one of history's most notorious killers. But what about its cousins? These guys aren't hiding in the shadows. They are literally part of the environment we live in every single day. And trust me, they have their own fascinating story to tell.
So what exactly are NTM? Well, the name kind of gives it away, right? They are all the mycobacteria that are not part of the tuberculosis family. You might also hear them called atypical mycobacteria. And this isn't just a handful of bacteria, it's a hugely diverse group of organisms found naturally all over the planet in soil and water. And this is where it gets really important to draw a clear line between the two. Tuberculosis is what we call an obligate pathogen. It pretty much needs a human host to survive and spread. NTM, totally different. They're perfectly happy living out in the environment. And that leads to the single most crucial difference. TB is contagious, it spreads from person to person. NTM are opportunistic. They don't spread between people. They cause an infection when they find an opening, usually in someone with weakened lungs or a compromised immune system.
Okay, so you've got dozens and dozens of these different NTM species out there. How on earth do scientists even begin to keep track of them all? Well, as it turns out, they came up with a pretty clever and surprisingly colorful system to bring some order to the chaos. Let's rewind to the 1950s. A microbiologist named Ernest Runyon was staring down this exact problem. He needed a simple, practical way to group all these bacteria. And the system he came up with was so effective, it's still a cornerstone of how we think about them today.
And here's the beautiful part. It's incredibly simple. He just looked at two basic characteristics. First, do the bacteria make any color, any pigment? And if they do, is it all the time or only when you shine a light on them? And second, how fast do they grow in the lab? Is it slow and steady, or are they rapid growers? That's it, a really elegant framework. So let's break this table down. Group I, the Photochromogens. Photo means light, right? So these guys only produce a yellow-orange pigment when they're exposed to light. A classic example is M. kensai. Then you've got Group 2, the Scotochromogens. These produce pigment even when they're grown in the dark. Group 3 is easy, the non-photochromogens. The name says it all, no color at all. And this group includes the really important MAC complex. And finally, Group 4, the rapid growers. These guys are just fast, forming colonies in less than a week, while the others can take weeks to grow. All right, so that's how scientists organize them in the lab. But what does all this mean for us, for our health? Let's get into the real-world impact of these environmental bacteria. Generally, when NTM do cause an infection, it falls into one of four main buckets. By far the most common are pulmonary infections, which affect the lungs. But they can also pop up in lymph nodes, on the skin, or in the most serious cases, spread all through the body in what we call a disseminated infection. Now, here's a great example with a really memorable name. Swimming pool granuloma. This is a skin infection caused by a species called M. marinum. It usually happens when someone with a little cut or a scrape gets into contaminated water, like in a swimming pool, or pretty commonly, a fish tank. And here's another one, scrofula. Sounds kind of old-timey, doesn't it? Historically, this neck lymph node infection was caused by TB, but today it's actually more often linked to NTM, specifically M. scrofulaceum, and we see it most often in kids. And we absolutely have to talk about MAC. This is a big one. It stands for the MAC avium intracellular complex. For most healthy people, it's not a huge threat. But for someone with a severely weakened immune system, like a patient with advanced HIV, MAC can be a total disaster, causing a devastating body-wide infection. It's a huge reason why NTM are taken so seriously. Okay, so imagine a patient comes in with symptoms. How do doctors figure out if it's a common bug, the dangerous tuberculosis, or one of these trickier NTM species? Honestly, the whole diagnostic process is like a great piece of detective work. And here is the central mystery they have to solve. When you do a standard lab stain and peek under the microscope, both NTM and M. tuberculosis look exactly the same, these little red rod-shaped bacteria. So if you can't tell them apart visually, how do you identify the real culprit? Well, the path to the answer follows these crucial steps. First, you've got to collect a specimen, like sputum from the lungs or maybe a tissue sample. Then, microscopy confirms you're dealing with some kind of mycobacterium. But the really critical next step is culturing it, actually growing the bacteria in the lab, because only then can you move to the final step, figuring out exactly which species it is. And here it is, the key clue that can crack the case wide open and fast. There's a rapid test for a protein called MP64. Here's the deal. This protein is only made by the bacteria in the M. tuberculosis complex. So if the test is positive, you've got TB. If it's negative, you're almost certainly looking at an NTM. It's a total game changer.
But it doesn't stop with that first clue. Modern labs have this incredible toolkit. They use things like PCR, which is basically a DNA photocopier, to hunt for specific genetic fingerprints. They can even use really advanced tech like mass spectrometry to get an exact ID on the NTM species. And that is absolutely vital, because that tells you which drugs are most likely to work. And that brings us to our final section, treatment. Because identifying the NTM is one thing, but actually getting rid of it, that presents a whole new set of challenges. This quote right here really says it all. The standard drugs that work so well against tuberculosis, most of them are pretty much useless against NTM. These organisms are just naturally tough customers, with built-in resistance to a lot of our go-to antibiotics. So because they're so tough, you can't just throw one drug at them and hope for the best. That's a recipe for failure. The strategy is always multi-drug therapy. You hit them with a cocktail of different antibiotics to attack from multiple angles. This is so important for preventing the bacteria from developing resistance and coming back even stronger later on.
So what does one of these drug cocktails actually look like? Well, a very common regimen, especially for big players like M. cansii or M. kansasii, combines 3 different drugs: a macrolide like clarithromycin, another one called ethambutol, and a rifampin like rifampin. This triple threat approach is really the backbone of modern NTM treatment. So when you step back and think about it, these NTM are just a constant part of our world. They're everywhere. The fact that they usually only cause disease when our own defenses are down, it really makes you wonder, doesn't it? What do these opportunistic bacteria really teach us about the delicate, invisible balance between our own bodies and the environment we all share?